Finally, we're ending on a 6 marker. However, it's quite a nice one. The student grew the bacteria on agar jelly in a petri dish. Describe each of the steps required for the student to prepare an uncontaminated culture and explain why they are examples of aseptic techniques. Now, this 6 marker comes up year after year. It's a really easy one actually because you just need to think through logically and clearly what you did in this practical. You need to cover 6 of those points, link it to the importance, and you'll get 6 marks. Firstly, you need to use disinfectant on the work surface to kill any unwanted pathogens already there. So that might be a spray, wiping the desk down, that sort of thing. Then you need to wash your hands thoroughly to prevent microbes on your hands getting into the equipment. Obviously, you're touching everything, that can't happen. You could tie your hair back to prevent hair falling and contaminating any cultures. The petri dish and agar jelly should be sterilised before use. This should probably be done for you by your lab technician, and that will kill any unwanted microbes on the equipment before you use it. Now, a really important one that you probably remember doing is passing the inoculating loop through a Bunsen burner flame, and that will kill any microbes already on the equipment. This is sterilisation. You then use that same inoculating loop to spread the bacteria onto the agar gel. The proper name for this is a bacterial lawn, and you might remember doing a little zigzag pattern on the gel. When you do this, you need to open the lid as little as possible to limit microbes entering from the air. So you just open it very, very slightly for as short a time as possible, and that limits the contact that the agar jelly makes with the surrounding air. The air is full of pathogens. After this, you need to seal the dish with sellotape to prevent microbes from the air entering whilst you're incubating it. However, you've got to be really careful that you don't completely seal that dish, so you don't make it completely airtight. Some oxygen needs to be allowed in, otherwise you'll get very, very dangerous anaerobic bacteria growing. Finally, you need to incubate your bacteria, like we talked about before, at 25 degrees roughly, and that allows a good moderate growth rate of bacteria so that you can look at it in a week's time, a few days' time, and see what's growing.